Welcome to another deep dive. We're tackling a study that's generating some serious buzz in the world of transplant infectious disease. HIV positive to HIV positive kidney transplants. Sounds pretty straightforward, right?
Well, not so fast. There's a lot to unpack here.
That's why you're here, to help us make sense of it all.
Absolutely. And this study, just out last month in the New England Journal of Medicine, is really shaking things up.
Okay, color me intrigued. So why is this study such a big deal? What makes it so groundbreaking?
Well, for a long time, these transplants from HIV-positive donors to HIV-positive recipients were completely banned in the U.S.
Right, I remember hearing about that, like totally off limits.
Yeah. It wasn't until 2013 with the HOPE Act and then research guidelines a couple years later that things started to change. And even then, these transplants were only allowed for research purposes.
So this study we're diving in today is a part of that ongoing research effort, trying to answer that big question. Can we make these transplants standard practice? Are they safe enough? Effective enough?
Exactly. The Department of Health and Human Services is actually looking at the data as we speak to decide whether or not to move these transplants from research into clinical practice.
Wow, so this study has some serious real-world implications. I can see why everyone's paying attention. Before we get into the nitty-gritty, can you give us a quick overview of the study itself? What exactly were they trying to figure out?
It was a non-inferiority observational study. Basically, they wanted to see if transplants from HIV-positive donors were at least as safe as those from HIV-negative donors.
Got it. And they were specifically looking at HIV-positive recipients, right?
Right. The idea is if we can show these transplants are at least as safe, then we can potentially open up a whole new source of organs for HIV-positive patients.
Makes sense. And that could be huge, right? Because there's such a shortage of donor organs.
Absolutely. And for HIV-positive patients, the wait for a kidney can be even longer.
So tell me a bit more about the participants in this study. How many people were involved? Where did they come from?
The study enrolled 198 HIV-positive recipients from 26 different transplant centers across the U.S.
26 centers. That's impressive. So not just one hospital or research institution.
Exactly. They wanted to make sure the findings were applicable to a wide range of settings. And out of those 198 recipients, 99 received a kidney from an HIV-positive donor and 99 from an HIV-negative donor.
Okay, so two groups of 99, one with HIV-positive donors and one with HIV-negative donors. Pretty straightforward so far. But now I'm curious about the how.
Yeah.
How did they actually structure this study? Because I imagine it gets a bit tricky when you're dealing with organ transplantation. You can't just randomly assign people to receive an organ from one type of donor or the other, right?
You're absolutely right. The National Organ Allocation System works in a very specific way, taking into account things like blood type compatibility, geographic location, and other medical factors. So you can't just say, okay, you get a kidney from an HIV-positive donor and you get one from an HIV-negative donor. It's much more complicated than that.
So how did they work around that limitation? How did they manage to compare these two groups if they weren't randomly assigned at the start?
Well, that's where the study design gets really clever. Are you ready to get into the nitty-gritty of the methodology? Instead of randomizing who got what kind of kidney, they had participants consent to receive a kidney from either donor type, HIV positive or negative, whichever one became available first.
Ah, I see. So they were basically saying, we'll take whichever kidney comes up first and we'll track how things go.
Right. And they followed these patients super closely for up to 4 years, gathering data on all sorts of things: medications, hospitalizations, infections, lab values.
Wow, so they really went all in on the data collection.
Oh, yeah, they were super thorough, trying to get as much information as possible to really compare those two groups.
It sounds like a data lover's dream.
Really?
But you mentioned they couldn't randomly assign donors. So how do they handle the fact that those two groups, the HIV-positive donor group and the HIV-negative donor group, might not have been perfectly matched at the start? Like, what if one group ended up with more patients who were older or had other health conditions?
That's a great question and a really important one. And that's where the limited observation arm comes in.
Okay, that sounds intriguing. Tell me more.
So they took some of the recipients in the HIV-negative donor group and randomly assigned them to this limited observation arm.
I am still not quite following. What did that actually do?
It was a way to kind of balance things out by controlling for some of those unavoidable differences that come up when you can't randomly assign donors.
So it was a way to make the comparison between the two groups more fair?
Yeah.
Even though they couldn't control who got which type of kidney? Okay, that makes sense. I'm always impressed by the clever ways researchers find to get around these kinds of limitations. So they collected all this data over 4 years. But what exactly were they looking at? What were the specific things they were tracking to assess how safe and how effective these transplants were?
Well, like I said, it was pretty comprehensive. They were looking at things like what medications people were taking, whether they needed to be hospitalized, any infections that cropped up, their lab values.
Got it. So all the usual suspects when it comes to monitoring a transplant patient's health.
Right. But they also did some things specific to the transplant itself, like allograft biopsies.
Allograft biopsies. Remind me what those are again.
Basically, it's a procedure where they take a small sample of tissue from the transplanted kidney to see how well it's doing, how healthy it is.
Okay, so they were literally peeking inside the kidney to make sure everything was running smoothly.
Yeah, they did those biopsies at set times, and also whenever there was a clinical reason for it, like if they suspected there might be a problem.
Makes sense.
Yeah.
And I imagine they were also keeping a close eye on the recipient's immune system
Absolutely. They did donor-specific antibody testing.
to check if the recipient's body was starting to make antibodies against the donor's kidney, which could lead to rejection, right?
Exactly.
Wow, so they were really taking a holistic approach, not just looking at survival, but at the whole picture of the patient's health and how well the transplanted kidney was functioning.
Yeah, you got to look at the whole enchilada to really get a sense of how these transplants are performing.
And they did all this over 4 years. That's a pretty long follow-up period for a study like this.
Yeah, it gives us a much better picture of the long-term effects compared to, say, just looking at the first year after transplant.
Absolutely. So they collected all this data, followed these patients for 4 years, and now the big question is, what did they find?
Drum roll, please.
All right, lay it on me. What did they find? Was it safe? Was it effective?
Well, the big news is, yeah, the study found that kidney transplantation from HIV-positive donors to HIV-positive recipients was non-inferior to transplantation from HIV-negative donors.
Okay, that's the headline. But remind me again, what does non-inferior really mean?
It means that, statistically speaking, the risks were no worse for those who got a kidney from an HIV-positive donor compared to those who got one from an HIV-negative donor.
So they met that benchmark. But I'm sure there's more to the story. What were some of the specific outcomes they were looking at, and how did the two groups compare on those?
Oh, yeah, they looked at a whole bunch of stuff. And like I hinted earlier, some of the findings were pretty surprising, even for us seasoned transplant ID folks.
All right, well, don't leave us hanging. Give us the details. Let's start with the big one, survival. What did they find there?
Well, both overall survival and survival without graft loss were very similar between the two groups.
Okay, so that's reassuring.
Yeah. At 1 year, 94% of those with HIV-positive donors were still alive, compared to 95% with HIV-negative donors. And at 3 years, survival was 85% and 87% respectively. Pretty much a wash.
And what about graft survival specifically?
Also very similar. At 3 years, 84% of those with HIV-positive donors were still alive with functioning grafts, compared to 81% of those with HIV-negative donors.
Wow, so whether you got a kidney from an HIV-positive or HIV-negative donor, your chances of survival and your graft's chances of survival were essentially the same. That's huge.
Absolutely. It really challenges some of the preconceived notions about the risks of these transplants.
For sure. Okay, what about rejection? Any difference there between the two groups?
This is where it gets interesting. You might think that getting a kidney from someone with HIV would lead to higher rates of rejection, right?
Yeah, that would make sense intuitively.
Well, this study actually found the opposite. Lower rejection rates in the HIV-positive donor group.
Wait, really? That's not what I was expecting at all.
Yeah, it was a bit of a head-scratcher at first, but it seems like the higher use of ATG induction therapy in this study played a big role.
Right, that super aggressive immunosuppression to prevent rejection early on.
Exactly. 66% of the participants in the study received ATG, which is higher than what you typically see in other studies.
So that might explain the lower rejection rates in the HIV-positive donor group. But it also makes you wonder if maybe we should be rethinking our induction protocols across the board, even for HIV-negative to HIV-negative transplants.
Right. It's a fascinating question that this study raises. Maybe more aggressive induction therapy could benefit everyone, not just those receiving kidneys from HIV-positive donors.
Absolutely. So we've covered survival, graft function, and rejection. What about the risk of opportunistic infections? That's always a big concern for transplant recipients, especially those with HIV.
Definitely. And it was a major focus of this study. The good news is the rates of opportunistic infections were similar between the two groups, and even more encouraging, they were lower than what we see in previous studies of HIV-positive to HIV-positive transplantation.
Okay, so comparable rates between the two groups, and even lower than historical rates. That's promising.
It is. CMV and esophageal candidiasis were the most common infections they saw, but overall the rates were relatively low.
That suggests that with good management, with appropriate prophylaxis, those infections may not be a significantly increased risk in this population.
Right. It's all about careful monitoring and proactive management.
Now let's talk about the elephant in the room, the thing that probably makes people the most nervous about these transplants, HIV breakthrough infections. What did the study find about that risk?
Okay, so here's the deal. The study did find a higher rate of HIV breakthrough infections in the HIV-positive donor group. However, and this is a big however, almost all of these cases were linked to people not taking their antiretroviral therapy as prescribed.
So it wasn't necessarily the transplant itself causing the breakthrough, but rather a challenge with medication adherence.
Exactly. And that's something we need to be really diligent about addressing in all transplant recipients, not just those with HIV.
Absolutely. Making sure people understand the importance of sticking to their medication regimen even when they're feeling good.
Right. It can be a matter of life and death, literally.
Okay, one last thing I want to touch on before we wrap up HIV superinfection. Can you explain what that is and whether it was a concern in this study?
Sure. Superinfection basically means that someone who already has HIV gets infected with a second, different strain of HIV from the donor. And I want to emphasize that this is extremely rare.
Okay, good to know.
The study only found one potential case of superinfection out of the 99 recipients of kidneys from HIV-positive donors who had genetic testing done. And even in that one case, they couldn't definitively confirm that it was actually superinfection.
So incredibly rare and not even a confirmed case in the study. That definitely puts things in perspective.
For sure. And I think it's important to highlight these nuances because, when people hear terms like superinfection, it can be scary. But the data here suggests that the actual risk is really, really low.
That's good to hear. So we've covered a lot of ground today. Survival, graft function rejection, opportunistic infections, HIV breakthrough, even superinfection. And the data seems pretty darn compelling.
It does. This study really moves the needle forward in our understanding of HIV positive to HIV positive kidney transplantation.
Absolutely. I think it's safe to say that this research is going to have a major impact on the field, potentially changing the landscape of transplantation for HIV-positive patients.
I agree. It's a real game changer.
Well, on that note, I want to thank you for taking the time to break all of this down for us. It's been a fascinating deep dive into some truly groundbreaking research.
My pleasure. Always happy to geek out about transplant ID.
And to our listeners, thanks for joining us. We hope you found this deep dive informative and thought-provoking. We'll catch you on our next deep dive into the cutting edge of transplant infectious disease.